Welcome to this video on glycogenolysis, an important metabolic pathway that helps maintain normal blood glucose levels during fasting and provides glucose to vital tissues. In this video, we will define glycogenolysis, explore tissues where it occurs, and presents its mechanism, key steps, enzymes, regulation, and clinical significance. Glycogenolysis is the metabolic pathway that releases glucose 1-phosphate from the non-reducing ends of glycogen through enzyme action. Glucose 1-phosphate is converted to glucose 6-phosphate, which is used locally by a skeletal muscle for energy, or converted to free glucose by the liver, which supplies it to other tissues. To understand glycogenolysis, we will examine the basic structure of glycogen and identify its non-reducing ends. Let's explore the site of glycogen storage and the sites where glycogenolysis occurs. The liver and skeletal muscle are the main glycogen storage sites in our body. The liver stores about 100 grams of glycogen, which is roughly 8 to 10% of its weight. This glycogen is a stored energy fuel that supplies glucose during fasting for up to 12 to 18 or 20 hours. Skeletal muscle, on the other hand, stores around 400 grams of glycogen, which is 1 to 2% of its weight. Muscle glycogen, however, is used locally for energy during activity and does not contribute directly to blood glucose because muscle lacks glucose 6-phosphatase enzyme. These organs, liver and skeletal muscle, are also the sites of glycogenolysis. To understand the mechanism of glycogenolysis, it is important that we understand the basic structure, reducing ends, and the type of bonds in a glycogen molecule. Glycogen is a homopolymer of alpha-D glucose units connected together with alpha-1,4 glycosidic bonds shown in red lines, and the branching is formed by alpha-1,6 glycosidic bond, which is shown here with blue lines. The multiple points or the ends which are shown here with the green color are the non-reducing ends from which glucose is released during glycogenolysis by the enzyme glycogen phosphorylase. Before explaining the mechanism of glycogenolysis, I would like to give a brief review of the enzymes involved in this process. The first enzyme is glycogen phosphorylase, which releases glucose from glycogen at the expense of inorganic phosphate. It produces glucose 1-phosphate and therefore it saves cells energy. It also utilizes pyridoxal phosphate as a coenzyme in this reaction. The second enzyme is debranching enzyme. This is bifunctional enzyme. This has transferase activity and second activity is alpha-1,6 glucosidase. Third enzyme is phosphoglucomutase. This converts glucose 1-phosphate to glucose 6-phosphate so that it can be utilized in glycolysis. The last enzyme is glucose 6-phosphatase, which is present in liver to release free glucose so that it can be distributed to other body parts. However, this enzyme is absent in the skeletal muscle. So let us examine the mechanism of glycogenolysis. This is a glycogen molecule having two branches which are connected with alpha-1,6 glycosidic bond, and this end is the non-reducing end. This is the end glycogen phosphorylase start chewing and producing glucose 1-phosphate. However, there is a limitation for this enzyme. When it reaches 4 glucose unit away from the branch point, it is stops working and therefore it produces glycogen phosphorylase produces glucose 1-phosphate and limit dextrin, the remainder of the glycogen. This is produced by phosphorylase and this is resistant to glycogen phosphorylase, means it cannot be hydrolyzed any further unless this hurdle or this branch point is removed here. So this is removed by transferring these 3 glucose unit to the other branch by debranching transferase activity, thereby extending the chain so that phosphorylase can start working again. However, because the branching is still intact it must be removed, otherwise phosphorylase will stop again. And to do that, debranching enzyme glycosidase activity remove this glucose as free glucose and linear glycogen molecule, which is hydrolyzed subsequently with glycogen phosphorylase producing glucose 1-phosphate. In the next slide, we will learn how glucose 1-phosphate and free glucose are utilized by liver and skeletal muscle. Glucose 1-phosphate, which is produced during glycogenolysis, is utilized by liver and skeletal muscle by converting it into glucose 6-phosphate by enzyme phosphoglucomutase. Glucose 6-phosphate subsequently is converted into free glucose in liver with the help of glucose 6-phosphatase enzyme, and subsequently this is distributed to other body part or tissues. However, in skeletal muscle, glucose 6-phosphatase enzyme is absent. Therefore, glucose 6-phosphate is directly utilized in glycolytic pathway for energy needs, and skeletal muscle therefore cannot contribute to blood glucose level. During glycogenolysis, we have observed that debranching enzyme releases a small amount of free glucose from glycogen branch points. Does skeletal muscle release this free glucose into the blood? The answer is no. In skeletal muscle, hexokinase converts free glucose into glucose 6-phosphate, thereby trapping it inside the cell. And because muscle lacks glucose 6-phosphatase enzyme, it cannot release and share free glucose into the blood. Regulation of glycogenolysis. Regulation is partly similar and partly different in the muscle and liver. For example, glucagon activates this process only in the liver, whereas it has no effect in the muscle because muscle does not express glucagon receptor. On the other hand, epinephrine activates this process in both organs, liver and muscle, through cyclic AMP dependent pathway. In addition, AMP and calcium ion activate this process only in the muscle. They have no effect in the liver glycogenolysis. On the other hand, ATP, high concentration of ATP, insulin and glucose inhibit this process in the liver, whereas only ATP and insulin inhibit this process in the muscle. Glucose has no effect in muscle glycogenolysis. Glycogenolysis is clinically very important because deficiencies in its enzymes lead to a group of disorders known as glycogen storage diseases. These conditions can affect multiple organs, a few organs, or specific tissues depending on the enzyme involved. For example, von Garke is associated with glucose 6-phosphatase deficiency in the liver, and therefore it is associated with hypoglycemia. Pompe, on the other hand, is associated with deficiency of acid maltase in multiple organs. Cori, on the other hand, is associated with deficiency of debranching enzyme in liver and muscle. Therefore, this condition is associated with symptoms of hypoglycemia and exercise intolerance. McArdle, on the other hand, is associated with glycogen phosphorylase deficiency in the muscle, and therefore there is muscle weakness as well as exercise intolerance. On the other hand, Hirsch disease is also associated with glycogen phosphorylase, but in the liver. Liver is affected here, and therefore these patients show symptoms of hypoglycemia. Thank you very much for watching this video. Please like it, share it and subscribe it and write comments. Until next time, you take care. Bye bye.